And yeah, so what we want to talk about tonight is what healthcare careers are available directly after Bowdoin, and have a chance to actually speak to some alums who are doing these jobs right now. So first, let's go ahead and meet our fabulous alumni. We have Emma Berkey, class of 2020, majored in biology and is now a clinical research coordinator at Dana-Farber Cancer Institute. And these are in alphabetical order. I'm not showing any favoritism. I just want you to know. So, and then Gabriel Quillian, class of 2023, majored in chemistry and is a post-baccalaureate researcher at the Hospital for Special Surgery. We have Meredith Flynn, class of 2017, who is a biochem major and is now a clinical research manager at Dana-Farber Cancer Institute. We have Liz Gracie, who is class of 2022, majored in biology and is now a clinical research coordinator too at the Center for Mindfulness and Compassion. And we have Omar Gunjo, who is in the class of 2021, also a biochem major, and is a research technician at Fred Hutchinson Cancer Research Institute. Or Research Center, I apologize. And then I am not an alum, but I did want to introduce myself. My name is Erin Beirne. I'm the advisor for science and healthcare careers here at CXD. So if you haven't come to see me already, please do. We can talk about all things internship, jobs, food, dogs, and etc. So let's see. So I am interested to hear from you guys about what kind of a typical day is like in your role. And I know that that's a hard question to ask because there's probably a lot of variety, but you can kind of take us through that in whatever way you would like. So, and then I'm just going to call on folks. So could Gabriel, would you mind taking us through your typical day?
Excuse me. So my typical day here, I work here in New York City, Hospital for Special Surgery, and it's a basic research lab, and we work with understanding the bone biology and stem cells that differentiate into osteoblasts. And so we do a lot of our research through mice. And so I take care of the mice. And so my typical day would be going down to the mouse room and checking up on my cages and making sure that some of the mating cages, see if we have any new litters with new pups. And if they're old enough, we like to tag them and genotype them because we want to see how the genes interact with differentiating these osteoblast cells. And then a lot of my time is spent genotyping, but I also do a lot of histology. So I section the femur and the muscle surrounding it, and I will stain it for specific antibodies and look at it under a microscope and see how it differentiates into like specific cells, osteoblast cells, muscle cells, and things like that. I'm in a small lab, so I have to take care of half of everything that's being done.
Wow, that's amazing. And thank you for kind of starting us off with what your position is and kind of getting us oriented to where you are. That's great. So Liz, would you be willing to go next?
I'd be happy to. So my daily duties have changed a lot over the year and a half that I've been out of Bowdoin. And that's because I work for a center that whips out a lot of grants, and we're working on a lot of projects. So it's a really fast-paced environment. I work at the Center for Mindfulness and Compassion in Cambridge, Massachusetts. It's out of Cambridge Health Alliance, which is the teaching hospital for Harvard Medical School. And the center is led by a principal investigator who is an addiction psychiatrist. So we're looking specifically at opioid use disorder and the ways that mindfulness can work as a treatment for opioid use disorder, and specifically Suboxone retention. I was working on that study for a while, which meant I was consenting participants, being HIPAA compliant, working with the IRB, filing adverse events. I would facilitate mindfulness classes at night. So my day would sometimes start at 10 and end at 7 or 7:30. And that was I am so grateful for that participant facing work, because in the long term, I hope to be a clinician. So being able to work and use motivational interviewing to help create a comfortable space for people in a vulnerable population was very meaningful. Since then, I have transitioned to be full-time on a study that I'm leading, and it's on looking at the adverse health effects of opioid overdose-related bereavement. So we're looking at substance use outcomes and mental health outcomes. And with that, I've been doing a lot of presenting, like poster presentations, getting funding from private donors, applying for an R34 NIH grant, and we'll be applying for an R01 in June. So that's a lot of work. And right now I am working on my first primary author manuscript. So it's been a lot of meetings and planning. I've had to learn R and code and do surveys. So I think Bowdoin uses Qualtrics, but our center uses REDCap. So it's really everything from writing to HIPAA compliant participant data information and security, presentations, and knowing other people in the field. So I've been able to recruit a cohort of study team members for the R34 and the R01, and it's been very fun because it's all the people that you have been reading about, all the manuscripts that in college that you get to read, you then get to work with these people. So yeah, I guess that wasn't a very eloquent answer of what happens in a day, because it's always changing and a lot of Gantt charts.
That's wonderful. I have one quick follow-up for you. And so how long have you been there as well?
I started at CMC 48 hours after I graduated, which I really don't recommend. So it's been about a year and a half.
Wow. That's an amazing kind of evolution over that time too. That's really cool.
Yeah, thanks.
Yeah, so Meredith, would you be willing to share with us what your kind of typical day looks like?
Yeah, sure. So just to give a little background, I actually started in the same role that Emma is in now, as a clinical research coordinator. So I'll give a flavor of what that looked like. And then I'll talk a little bit about what I do now. So right after Bowdoin, I also started 2 days after graduating from Bowdoin. Again, don't recommend that. I started in the genitourinary department at Dana-Farber. So one thing to note, Dana-Farber is very separate based on disease and department. So role is not necessarily exactly the same across departments. So it's a good question to ask in an interview to know the difference. So my role as a CRC was on the therapeutics team or treatment team. And I worked mostly with prostate cancer patients. So everyone that I was working with was an older man and was diagnosed with prostate cancer. I worked on mostly trials that patients had many different treatments, and then we're on a trial to try to see what other options were available to them. So my initial role was coordinating the trials. I had a portfolio of trials that I was a lead clinical research coordinator on. I did all of the patient prep. I worked with the scheduling team to book their visits according to the protocol. And then I saw my patients in clinic, made sure that they were having their visits align with what the trial needed. to ultimately get the data out at the end. And I was the one entering the data and doing AE reporting, as Liz said, and adverse event reporting, and submissions to our IRB, and then coordinating very closely with the pharmaceutical companies or the funding sponsors for these trials. So that was a little bit of my day-to-day as a CRC. Very collaborative environment. Many people are coming directly from the undergrad world into this clinical research coordinator role. So honestly, it was a really great transition from undergrad life to, moving into a different city and different role. I stayed in that position for a few years and then decided that, I was looking for what was next, and decided to stay on actually in an assistant clinical research manager role for the same team that I was on. So moved into management at that point, and then May of 2020, so early pandemic, the person ahead of me told me they were leaving, which pushed me into the CRM role, or clinical research manager role that I'm in now. My role now is essentially managing the team that I previously was on. I help onboard, train, hire. I do portfolio management, work very closely with all of our principal investigators, and meet with sponsors to make sure that we're equipped for their trials, manage deadlines, many different things. But I'm still very much connected to the role that I had post Bowdoin, which I feel like is pretty rare, being 6 years out at this point. But Dana-Farber's been a great place for growth. So really happy about that. Hope I answered everything. It's hard to say one day. This is what I always answer in an interview. There's no typical day, which is my favorite part, that every day is different. They're always learning something new. I still learn every single day something new, even though I've been here 6 years.
That's wonderful. And yeah, really interesting to hear about how you've kind of changed over time, and the fact that, the different departments are pretty distinct from one another. And so you can ask some good questions in interviews about that, or should. Yeah, so I guess kind of following up on that, Emma, would you like to share with us a little bit about your role?
Yeah, absolutely. I think Meredith touched on a lot of great points, but like she mentioned, between the different medical departments, there are a lot of differences. So I'm within the bone marrow transplant team, and one of the key differences between the kind of role that Meredith was describing, I would say, is that we don't actually have a lot of patient contact. Like, we personally do not typically meet with the patients in clinic unless a few of my colleagues have trials where they do consent the patients directly, but important for people who are applying to jobs and really wanting that sort of face-to-face patient contact to maybe just do a little bit of extra research, because even across departments that sort of thing can change. And I have been in this position for 5 months, so more of a recent addition to the team, but I've been really enjoying it so far. Like Liz and Meredith both said, I don't think that there's a very standard typical day. I was trying to reflect over on how to distill the different things that I do, and just was thinking of today as a possible example. One of the first things that I did when I came in was picking up a consent, some data entry, enrolling a patient. There's a lot of email coordination across all different people of the medical team, which always ends up taking a large component of the day, scheduling. And then thinking of even just my different teammates, I know some of them do far and away just data entry for 99% of their day. And then I have other teammates who do a lot of in-person active coordinating samples, packaging and shipping samples. So I think it really can widely vary, and adverse event reporting, of course. But yeah.
Wonderful. Thank you so much. And Omar, would you be willing to tell us a bit about your typical day?
Of course, I'd love to. I work in, I'm a research technician in the lower back lab at Fred Hutch Cancer Research Center. Our lab studies proteostasis, which is basically how your cells regulate protein folding, and how they get rid of misfolded proteins and aggregates and stuff like that. And it's a basic sciences lab. we use C. elegans, which is a tiny nematode, like a worm species, to do a lot of our research. And it's mostly like genetic research. On a typical day, there's always traffic on the freeway, so I spend an hour in traffic getting to work. But once I get here, like there's a lot of different strains that we use to like, probe the different research questions that we're trying to answer. So I do a lot of like strain maintenance. You know, the worms, like they have a short lifespan. They lay about 300-ish eggs in a few days. And so like when I come in, I pick like a new worm onto a new plate to like maintain that specific genotype. So I do strain maintenance. Uh, I do a lot of like crossing assays, like mating assays, to get like one genotype of one strain and combine it with another genotype to like use it in experiments. I also do a lot of, like Gabriel, I do a lot of genotyping to like confirm that like, okay, this strain that I just made has the genes that I want and they're like homozygous and whatnot. I guess typically in the day I'd set up like experiments to maybe like expose certain strains to like, an inhibitor drug to like investigate something specific like that. So my day to day is like very bench work heavy, a lot of pipetting, a lot of like, you know, looking through a microscope to like pick worms or like looking at like fluorescence reporters and stuff like that. And yeah, I think I also do like I'm a research technician, so I help with like maintaining media, making sure we have like plates ready, making sure our solutions are fully stocked, ordering stuff. Yeah, I think that's a good overview.
That's great. Did you know when you were leaving Bowdoin that you wanted to do something that was very bench work heavy?
Yeah, yeah. Like I guess my goal was to try to like really immerse myself in the research environment and just get a sense of like, okay, what is it like to be a full-time researcher? Like what's the day-to-day like, kind of like going at this question myself, like what does a typical day look like? You know, yeah.
Yeah, that's great. Yeah, I mean, it's a hard thing to get a handle on while you're in undergrad. You have some shorter periods of time where you can do that, but yeah, that's great. Okay, so for all of our participants here, for our I guess audience, students, do you want to ask any questions of our panelists now? I'll just give you a moment to either raise your hand or turn on your video or send me a chat so that I can ask. Otherwise, I can keep going. Okay, so maybe while people are thinking up their questions, we can move on to how did you find the job that you have now, and what platform or platforms did you end up using in your job search? So Emma, would you be willing to answer that for us?
Yeah, absolutely. My way of coming to the Dana-Farber job was actually through a good friend, a fellow Bowdoin alum who had worked there for, I want to say 2.5 years. And I actually had a little bit of an atypical route into the position because I was previously working at an animal hospital and thinking about vet school. And as I started to pivot away from that, I was talking to this friend who was headed into med school and had a fantastic experience as a CRC at Dana-Farber. And she was like, okay, if you want healthcare experience, like I have the job for you. And she really wanted to help out her team. So I know she was really, really pushing it, but I couldn't be happier that I took it. So she was actually the one who sort of first told me about the role. And I had a lot of information about the role through her, and then she was able to sort of forward my resume along to the manager. And then the process went from there.
Amazing. Thank you. And I want all of the students listening to note networking. Networking, it's so important. And talking to fellow Bowdoin alums who have gone through a similar path. So definitely, be active on LinkedIn and on your networking. So just a little plug there. And Meredith, how did you find your first position out of Bowdoin?
Yeah, sure. Honestly, very similar. Networking. I knew people working at Dana-Farber when I was graduating. And I will say there's an online website for Dana-Farber postings called iCIMS. We can certainly get the link sent around. I think actually our HR department is connected with Bowdoin and often sends a list of our job postings. And I will say I'm almost always hiring. I've hired 10 people this summer alone. There's always positions. So, there's various roles, and I'm happy to talk about the other entry-level roles that we hire into. But how I found my role was, through someone I knew. I talked to them about their experience at Dana-Farber. I applied online. They messaged people internally saying, this person went into Bowdoin, similar path. And then had my interview. I actually physically came here for my interview, which was really helpful. Met the team, and then started this position right after.
Awesome, thank you. Yeah, we'll have to keep an eye out for the Dana-Farber positions. I think you're right that some of them do come to us through Handshake, but yeah, definitely sounds wonderful. So, and Omar, how did you find your position?
I knew I wanted to do like a research job. So I wanted to like live in Seattle like after graduating. So I sort of like did some research on the different institutions in this vicinity. And I went on like their websites and their like career pages. They usually have like a career page where people like put in job postings. And I just went through and like looked at the different positions in different labs and found stuff that I was interested in and sent out like a whole bunch of applications. And yeah, it's pretty, yeah, it was pretty like like not networking based, but like, yeah, I was just going online, doing some research and sending out applications for me.
That's great. I mean, again, it can be really helpful if you already know kind of the area that you would like to be in. And yeah, it's good to hear like kind of targeting those research institutes and just going through one by one, seeing what you can find. And Gabriel, how about you?
So similar to Omar, I knew I wanted to stay in New York City because that's where I was originally from and take advantage of going back home and saving money. So, and I've wanted to do orthopedic research. So, hospital for special surgery being number one in orthopedics for 14 years was the perfect spot. So, I did the same thing and I sent out my application to as many people who were hiring here at HSS as possible. I got in contact with some doctors who could probably network me in, and I think a lot of clinical research jobs, which is my priority at first, required a 2-year commitment. So I knew I needed to at least take the basic research position job because my goal was to only take a 1-year growth year. And so knowing how much time you want also helps because you can limit the jobs you can take. Obviously, you have more opportunities if you take more growth years, but if you're only taking one, sometimes learning the clinical side of it takes a lot more time and usually requires a 2-year commitment.
Yeah, that makes a lot of sense. And it's definitely a conversation I've had with a lot of students as they're looking and they're trying to think about, their pathway. So that's really good to hear. Liz, how about you?
Yeah, I also I didn't get my job through networking. I instead kind of spammed every hospital and university that I was interested in working with. So I started off by knowing what my main goal is, which is to build up my CV before applying for a PhD program, and a PhD in clinical psychology. So I wanted patient-facing work, that would allow me to try out a lot of different modalities of research. So then I go, okay, where am I willing to live? Because I believe that work-life balance is important, which I wouldn't say is like always prioritized in the healthcare setting, but I just knew where I would want to live. So I was looking at Boston, and then deciding would I prefer to be in a lab affiliated with a university, or would I want to be in a hospital setting? Um, and then after I like narrowed down that funnel a bit, I had a couple of criteria that I could look for, um, or that I could filter my search with. So then I really just like took to the internet, which might be bad advice, but looking at Handshake, Glassdoor, looking at specific hospitals. So I would look up MGH, clinical research coordinator positions available, um, or Beth Israel or McLean, and then just keep on applying. I think I sent in like probably like 35 applications and got 3 interviews. So my ratio wasn't great, but it just takes one yes. You just need one offer that feels right for you. It's yeah, you just need one opportunity that feels like a good connection, and then listen to your intuition with it. Yeah. Am I missing any part of the question? No.
No,
Okay.
I don't think so. No, that was great. It kind of took us through your thought process, which is really helpful. Um, So, and then I have a question from a student. And so they said, how did the panelists discover a passion for the specific research they are partaking in now? There's so much to choose from. And is this a stepping stone for them? Do they have even larger goals in mind? So yeah, Emma, would you mind if we started with you?
Yeah, of course. I might have to circle back and ask you the question again and make sure I hit every part of it. But starting with how I found my way to the specific kind of research that I'm doing. I did know, like I mentioned, I was sort of recommended this position from a friend. I knew that I wanted a position that would give me a lot of access and experience just sort of within the different roles of a hospital. So I knew I was very interested in working in a hospital environment, and looked at some different roles within that, which relating to another part of the question, which I think was what are bigger goals, as I pivoted away from thinking about vet school and working as a vet down the road, I realized that I was more drawn to human healthcare, and I'm now thinking about nursing school and then working within the field for a while, and then most likely going on to nurse practitioner once I am able to figure out which specialty is most appealing to me. But so for that reason, once I sort of figured out my graduate school direction, that was why I wanted to gain that experience just working with different clinical roles. And that friend who was leaving was really talking up her department. And I actually didn't know a lot about bone marrow transplant specifically before starting to work with them. So it would be disingenuous to say that that was the one department that really, really called to me amongst all the others. But I have learned so much about it since working with them. And I think that it's a really cool, like cutting edge kind of research, especially because the procedure of, a bone marrow
I am
transplant.
actually.
Sorry, go ahead.
Sorry, I think somebody was unmuted.
Unmuted. Okay, no worries. I was just gonna say, I think bone marrow transplant really cool because it's kind of a cutting edge field because the procedure was developed recently. So there's a lot that is still new and unknown and being developed. So retroactively, that's something that I would have found appealing. But I also definitely, similarly to, I think it was Liz who mentioned like work-life balance, I was really drawn to a hybrid role. And I think there were some roles within Dana-Farber that did not have that hybrid component. So along with that, they had more patient-facing opportunities, which some people might rank more highly in their priorities, and they would want more of that. But I did, because I knew I was going to be taking post-bac classes, I wanted a little bit more of the flexibility to be hybrid. And so that was something that the BMT department did offer that some others didn't. So that was definitely a big component for me as well. And then Erin, were there any other parts of the question that I didn't touch on?
No, I think that was great. Thank you.
Yeah, no problem.
Everything. So yeah, Omar, do you want to take a crack at it? So just kind of how did you discover your passion for this research, and do you have larger goals in mind?
Yes, I guess from the courses that I took in undergrad for biochem, I knew that I was interested in like cells and like the biology of cells and molecular like mechanisms of different things, but it was sort of like a broad interest and nothing specific. I remember in senior year, I took Professor Doobie's chemical biology seminar. I wonder if she's still teaching it, but yeah, I remember just, we read papers every week and like we had discussions about the papers, and I really like enjoyed like delving into like the specifics of those papers, understanding the methods that they use, understanding how they formulated hypotheses and went about answering their questions. And a lot of the papers were like dealing with stuff of like clinical relevance. So I really enjoyed that intersection of like biology and like, the clinical, like medical side of things. And like specifically the lab I work in studies proteostasis. And I was drawn to this lab specifically because I remember one of the papers we read in that seminar was about PROTACs, which is kind of like related to proteostasis. It's, a system that people use to like target the degradation of certain proteins. But I remember finding that paper pretty interesting. And when I was looking through the different positions posted on Fred Hutch's website, I saw like proteostasis and I started like reading up on it a little bit and looking at the PIs like publications and like kind of the different things that they look at. And it just, yeah, it felt like almost like a full circle moment. I was like, wow, this is like really cool that I'm actually seeing something that I learned about from class, like, in practice, in a larger research setting. And that kind of drew me towards applying for this job specifically, and like, spending time here as a research technician. I do have larger goals of going to medical school. I am like currently applying, currently in that anxious, phase of like, well, I sure hope they, send me like an interview invite, interview invite, just, patiently waiting and praying. But yeah, I plan to attend medical school in the long term. Did I miss any part of the question? Yeah.
No, I think you absolutely got there, and it was really interesting to hear kind of how your interest in a particular course at Bowdoin, like, led you right there. And it was great to hear about kind of some of the process of like digging into what that lab does and seeing like, does this feel like it would be a good fit for me? So that's awesome. And let's see. Liz, do you want to talk to us about that a little bit?
Yeah, I'd love to talk to you about that. I guess I had a similar experience of having a really good experience with Professor Reese at Bowdoin. I loved her abnormal psychology class, and I did an independent study with her looking at like an in-depth literature review about prolonged grief disorder. which was new in the DSM, the Diagnostic Statistical Manual for psychology, as of March 2022, which was my senior year. So I was really excited to do this literature review in an intervention proposal. And then in my interview at CMC, I was actually interviewing for the clinical research coordinator position for exclusively for this mindfulness and opioid use disorder study. But then when I mentioned my work at Bowdoin, looking at prolonged grief disorder, they kind of switched the offer and asked me to lead their research on grief related to opioid overdose. And I guess I want to preface by saying I didn't know anything about addiction psychology, or I actually never took neuro classes at Bowdoin, so I didn't understand how addiction worked. And so, it was kind of just like trusting my gut that I know I can learn quickly, and then I trust that my curiosity can lead me in the right direction. So then going to CMC and actually using my knowledge about prolonged grief and traumatic grief actually suited me very well. And I'm so grateful for Professor Reese for allowing me to have that opportunity with her. Yeah, and I guess like what I said before of just feeling a lot of gratitude, because I remember meeting with Hannah Reese to talk about a really interesting manuscript that I read. It was written by Holly Prigerson, who is kind of the reason why this disorder is in the DSM. And she actually just gave me feedback on a manuscript that I offered her co-authorship on. And it was really positive feedback. So it's like the shrine in my bedroom to Holly Prigerson is actually manifesting a professional relationship with her. Yeah, so things have fallen into place with a lot of hard work and manifesting. Yeah, I would just a piece of advice that I would provide is think about like what you're willing, like what are your bounds, like if patient-centered work feels uncomfortable, or I guess like being young and putting yourself in a position where like you're dealing with really heavy stress and themes of trauma, then just acknowledge and be aware of like what your capabilities are, because you don't want to be in a position where that clinical work feels like too much, and then it's no longer serving you or the participant. Yeah, but it's been a good ride for me so far.
Thank you. That's great to hear. It's, yeah. And all of these insights are so helpful, just to think about what are good questions to be asking yourself, during this process. And then, like during like looking for jobs, but then also like while you're there, and, is this continuing to, to meet these goals that I have and to serve me and to serve others. So Gabriel, do you want to address that question too, of how you discovered your passion for this specific research and your larger goals?
So I guess I don't have like specific interest in this research specifically, but in orthopedics, the field in general, I've come to grow an interest from my time as a prior athlete and visiting orthopedics myself and having to go to like the doctors for it. And I just wanted to, sometimes I didn't receive the best care, and I wanted to be that person, because a lot of people from underserved communities like mine try to use sports as a way out. And if they're not receiving the proper care, then they can't continue on that path. And so that was always my spark for orthopedics specifically. And similar to Omar, my future is medical school, and I'm in the cycle right now, which is kind of why I'm dressed up, because I just came from my first interviews. So yeah.
That's great. I mean, again, it's, yeah, you still have that passion and driving you towards this goal. And, even if it's not this specific thing that you're studying that you're passionate about, it is advancing you towards that goal. That's really cool. And I think Meredith, I don't know that I've gotten to you yet. I'm losing track, which is great.
All good. All good. I'll give a quick answer to this. I think I coming in had no idea anything about prostate cancer research. And I think, the big thing I would always say is you don't have to know what you're getting into. I think most of these positions you will learn on the job, and they will, you'll be with experts if you're at these hospitals. And so, definitely don't feel like you need to have some baseline understanding. I had a few internships while at Bowdoin that I really enjoyed, and knew that I enjoyed the oncology work, and I knew that there was so much room for growth in oncology, and room for research. And so that helped me think of Dana-Farber. But I don't think there was any, a specific reason that I, had a specific passion for this type of cancer research. It was more oncology in general. So don't put too much pressure on yourself in that way. I have learned to really, enjoy learning about the field. There's always so much moving and changing. And so if you like to learn, doesn't matter the subject matter, there's so much out there to learn. In terms of, whether this is a stepping stone, larger plans, coming into Dana-Farber, I knew already that I didn't want to go medical school route. But I knew I wanted something in healthcare. I came in thinking hospital administration, something more operational healthcare. Being here confirmed that I didn't want to do patient-facing role, which is partially why I moved into the management position, because I knew there was more I could learn at Dana-Farber, and this was a path outside of the patient-facing role, but still doing operations, clinical research, and having impact on patients without being, face-to-face with them. So I imagine myself, staying in either clinical research or healthcare operations on a larger scale, and really enjoy kind of this path of work that's less traditional than the pre-med track often tends to be. Though I will say my husband was pre-med at Bowdoin and went directly to medical school post Bowdoin and is loving that life too. So there's many different options. I will say that one of my big things I always talk to career planning about was that I didn't want my first role to close doors for other roles, and that was something that I was really hung up on leaving Bowdoin. And I have seen people go from a CRC role into being a lawyer. They've done PsyD, they've done MD, DO, NP, PA, nursing, you name it, they've gone to it. They've also had a lot of people stay internal, and a lot of people move into the pharmaceutical companies, or into smaller pharmas or startups. So I have seen people go into many different directions from the exact same starting point. So don't get too stressed about that either, because there's really so much you can learn here outside of even just the contents of how to be a CRC, that would set you up for success, in other roles. And I'm sure everyone would say the same about their position. You're learning the how to do day-to-day work, but you're learning so much more about, how a hospital works in a massive city, or how pharma interacts with the hospital. There's so many different ways to look at it.
Thank you so much. I know I keep using the word evolution, but it really is very helpful, I think, to hear about kind of how your thinking evolved over time, and, certainly the things that you were hung up on at the beginning, and, how has that changed? How did that manifest itself, once you got into that role? I think it's super valuable to hear. So thank you. And Meredith, you also kind of led us into another question from a student, and this seems like it probably will be the last one we'll have a chance to get to. And that's, so while you were at Bowdoin, did you have any jobs, internships, or experiences that you felt like helped to strengthen your application? And if so, how did you find those? So Gabriel, do you want to start us off with this one?
Could you repeat the question one more time? Sorry.
It's okay. We're throwing a lot at you. So while you were at Bowdoin, did you have any jobs, internships, or experiences that you felt helped strengthen your application? And if so, how did you find those?
So my research was, I did a research at Mount Sinai during my summers, and I also did a independent study with Manuel Diaz Rios. And I think having a lot of background in basic science research helped me get this job, because my principal investigator hired me because I had a lot of research experience, because he knew I was only going to be here for a year, but he knew that I could hit the ground running given my background. And because of that, I think those experiences really did help me. And for getting those positions, I think I emailed Manuel Diaz Manola, but it was before the neuro department started getting really popular, because he says that he's had to turn down a lot of research opportunities recently. And my research at Sinai was through networking, that someone that my mom knew connected me with the right person.
Awesome. That's great. And Emma, do you want to tell us a little bit about your experience?
Yeah, of course. During my summer before my senior year, I also did some research on campus, which led into an honors project with Amy Johnson in the biology department, working with sea stars. So not exactly directly related, sorry for any background noise, not directly related to clinical research per se, but still research related. Have a big love for marine biology as well. So I think I was just still exploring different areas, and it's hard to say if that exactly was what helped me, get this job. I think I had some work experience from a couple of years after Bowdoin as well, the animal hospital. And then I also worked with New York City Health and Hospitals for just under a year. So that could have helped as well. And similarly to Gabriel, I think I just reached out to Professor Johnson. I had a class with her, but definitely encourage anyone who's interested in a potential research route. It's great to get your hands dirty, and especially Bowdoin summers or just research during the year are a really wonderful way to do that.
Great, thank you. And Omar, how about you?
I also had a fair amount of like research experience during undergrad, and I think that really helped with my application. In sophomore summer, I did an internship at the University of Washington over here in Seattle. Uh, that was like my first like sort of serious research experience, just being there like every day and like, working consistently on like a specific project to try to get results. So that was very informative of research and the research process, and how like the real side of research, like it's not like you go from like A to B to C, like a lot of things go wrong and there's a lot of troubleshooting and optimizing and stuff like that. So that was very, very like informative. I also did a summer fellowship in junior year, which led into an honors project in senior year. And that also provided, valuable experience. And I think those combined helped strengthen my application for a job as a research technician, working in a lab full time. Yeah.
Great, thank you. Meredith, I know you told us a little bit about one of your internships, but do you want to tackle this one too?
Yeah, sure. I did 3 very different things after or during each summer. The first one was I went home and worked for my family's business, which was a grocery store, and was a cashier and worked making food, completely unrelated, but customer service, which actually I think is one of the things that I like to ask people about during interviews. So there's nothing that is not related. Um, then I'm trying to think back to make sure I get the order right, but I did an internship through the Daniel Hanley Center, which is a center actually I think based in Portland, and is working on connecting interns throughout the state of Maine. I had a really good experience there. I'm not sure if they're still connected. I think I found out about it through Bowdoin's website, honestly. I worked at an orthopedic office for that year or that summer. And it was a very small office. We had a lot of, they really let me kind of tailor to what I was interested in. I did a lot of report running for them. Was at the check-in desk some days. I really moved around and saw the different roles within the office, which I think gave me a really good scope of, how to run a private practice ortho office, though nothing related to what I do now. And then my third summer, I was wanting to go back home. I grew up in northern Maine, a few hours north of Bowdoin. And I reached back out to the hospital that was in the town that I grew up in and said, this is what I'm interested in, are there any internship opportunities? And they got back to me right away and said, come back. So that was, very nice of them. And I would assume a lot of places would do the same. I think people love to have, people go back home and, bring different ideas, even if it's just for a summer. So I worked on both the OB-GYN floor and they were opening an oncology center that summer, which just happened to work out timing wise. And one of my projects was trying to chat with patients to hear, what their preferences were while they were at getting their treatment. So was there a book they like to have? Was there a magazine type? Was there a television show? Did they like to have blankets? What was their go-to snack? Those types of things, which actually translate quite well into the coordinator role, because it was really about, getting to know the patient and understanding, that they're going through a tough time, but there are little things you can do to make it better. So those are my general experiences, one very through Bowdoin. I would also say I worked at the McKean Center while at Bowdoin and did a ton of volunteering and clubs that also were really valuable to talk about in my interview process as well. So highly recommend that too.
Excellent. Thank you so much. I think we should probably wrap it up there just because I want to be respectful of everybody's time. But thank you so much to our panelists. This was so fascinating to me, and I'm sure incredibly valuable for our students as well. Thank you.